right. Hello, everyone. Let's see. Not that many people in here yet, but that's fine. It's my birthday today. Hello. How you guys doing? All right. We are going to chat about let me pull this up. I did say it's my birthday. It's my birthday today, everyone. I am turning.
Happy birthday, Papa.
Thank you. I feel old, but now I'm in my mid-30s. I mean, maybe a little bit further into my mid-30s. All right, let's pull. But probably be celebrating a bit more tomorrow and over the weekend because I have still so much work to do.
What's your Gmail account? So that's what I needed.
Huh? What'd you say?
Oh, sorry, not you.
All right. Tomorrow I get to go give blood, which is good because I've got O positive blood and they want my blood. But all these places I was trying to get blood have canceled all of their drives. So at least it's something tangible I can give to this pandemic, right? All right. I can't drink too much tonight. We're gonna just have beer and pizza. It'll be fun. It's a chilly evening where I'm not in charge for dinner. All right. Sort of impossible to, have a big blow up party right now. Okay. Let's go. Assignments. See, I posted your assignment yesterday. It's a little bit easier to create. Sometimes it's daunting to put into the, what did we actually learn that is part of the actual assignment here. But I feel good about next week's because it's mostly what we learned today, and we'll learn a little bit of next week, okay? All right, hello everyone, great. Okay, let's pull up homework 9. Again, there is 11 homeworks. One of them is extra credit, so be sure to do all of them if you're interested in that. Okay, all right. We had a couple of problems that people got 70% on, so we will go through this. All right. Let's go to the student view. I will pull this all up. Okay. Let's share my whole screen on the next screen. Okay. Up to. All right. As I was saying, some of these look like you guys got it, so you didn't. So let's pull down. We can start on the one. This is difficulty 5. Median time. You took 10 minutes on this, so let's walk through that one. Give you a little bit extra points for that one, but let's see, make sure that everyone understands what's going on. All right, is this going to connect? There it is. All right. Whew. Yeah. So I know this is a lot of pieces here. I just wanted you to be able to put together all the parts. And I know that this is a lot of different parts here, okay? So I would have to look some of these up actually. Okay. RNA polymerase. Okay. Core protein is the protein without the sigma. Okay, so I can see how this is a lot of the pieces here. Okay, you had to look up what all of these different symbols mean, and so that did take a little bit of effort. I'm not going to be testing it like this on the test if you haven't taken it yet. I'm not sure anyone's taken it yet, but that's okay. You guys have until Saturday. So take a little bit to study because you only have one chance here, okay? So the key here is that you'd have to look some of these pieces up. But sigma, this is not sigma. This is sigma. Okay. Without the sigma. The active form of the enzyme is without the sigma, okay? I think this is a large enzyme. Now again, I would have to look up the actual pieces here, so we're gonna request the answer. But this one, again, this piece is here, okay? So it is the large, is the sigma, okay? The key here though is that this, so it's the alpha, beta, beta prime, and this W and that, okay? So I don't remember what the W does. So that's one of the things where, again, I was expecting you guys to look this up, so I can see how it would have been a little bit difficult, but that's the concept here, is that you had to use your book to look some of this up. Because we walked through a little bit of the structure in class. We didn't cover all of these pieces, but we did cover some of the parts here. The regulatory function we covered. So these guys we didn't cover that they are the catalytic mechanism, but that's the concept that is covered in the book. So this was mostly going over that piece. I won't quiz you on these terms though, okay? Except for here. All right, let's walk back for a second, make sure everyone has the concept here. All right. Okay, so who remembers this part? The mRNA formed from repeating tetranucleotide UUAC incorporates 3 amino acids, but the use of UAUC incorporates 4 amino acids. So what is remember this is part of the reading segment here. So what is the answer here?
Isn't it the second one that the triplets UUA and CUU produced?
Right.
Like they make the same amino acid.
The repeating tetranucleotide here means that as it repeats, you have UAA, and then after that would be CUU. But since UAA and CUU make the same thing, and then ACU makes a different one, right? Okay, so that's the 3 here, and then the UAUC does 4 because, again, as you read it, repeating it does that. But yeah, that's right. Has to do with the 3 reading frame here. Okay. All right, this one we talked about a lot. I think everyone got it. The methionine, right? Okay, so I'm not going to spend much time on this. It doesn't matter because I'm not getting graded on this. Okay. All right. Okay. Which of the following has the capacity to inhibit transcription?
All of them?
All
They
of them.
all do. All
Yes.
of them.
Right. The enhancer mutations, there's no enhancer, it can inhibit transcription. So if the enhancer is self-mutated, that's a problem. Methylated cytosines, those methylations can cut them off. The more methylated it is, the more cut off your actual chromatin is, the chromatin structure itself, or adding silencers. Again, all of those can do it. That's right. All right. Next. And then we talked about 3' and 5'. So what was the thing on the 3'?
Poly A binding protein.
That's a poly A binding protein, yeah. And the poly A polymerase makes it, but it's poly A binding protein actually that's on there. All right. So now we're talking about what mRNA does and how polyadenylation works. Okay. So what does it do and why is it there? Think about when it's added and why it's added. You can tell me one of them.
The first one.
First one, yes, longevity. It has to make it out of the cytoplasm. As I mentioned, it degrades pretty soon after it's read. So keeping it from degrading, because it is a very, very reactive molecule, keeping it from degrading is a big piece of it. Oh.
Next one, regulation of mRNA transport to the cytoplasm.
Yes. So this helps it to actually basically find its way, attaches it to the right cytoskeleton pieces, has to interact with proteins to carry it there. So yeah. What else?
Regulation of mRNA association with cellular components like proteins, lipids, or nucleic acids.
So this helps it to basically be recognized as an mRNA to the others. There you go. I think that's it, because here's the deal. Proofreading, that happens when it's being transcribed. Polyadenylation happens after it's transcribed. Intron length, that's all determined by the DNA. Splicing, not included with that A. Those should be the correct ones. Good job. You guys got it. All right. 3 post-transcriptional modifications. We already talked about some of these, but obviously the poly A tail. But where is the poly A tail? So there's a couple of places it says here, but where is this? What does it do? There's 3 things it does. So
5'
Kim says
capping. Yeah, the number 2. 5' capping, 3' A polyaddition.
Yeah, and splicing, exactly. Those are all have to be done before it can leave the nucleus. That's the maturation in eukaryotes. There's no real introns in bacterial DNA. All right, so the previous one hints at this one too. Most eukaryotic mRNAs are shorter than the genes that encode them because of which one?
They contain both introns and exons.
Yes. So note that you might be tempted to press this one, that introns are spliced out of the DNA, but it's not spliced out of the DNA. Yes, introns are spliced, but it's that exons are spliced. Exons are kept in, so these 2 are not correct in the way that they're described. All right. All right, alternative splicing. Talked about that a little bit in that the polymorphisms of genes. So, it's a lot of aberrant splicing, a lot of things, but what is the definition here of splicing? Start with part A.
It's
What's
the
the
first
definition?
one.
Say one more time.
The first one, yeah.
Alternative splicing occurs when copies of a pre-mRNA are spliced in more than one way, yielding various combinations of exons in the final mRNA products. Generally, in a functioning alternative splice, a purposeful one in that sense, in the sense that it's not a broken protein. It is where the exons are the ones that are alternately kept or not kept. And that's the kind of the relationship of that, the exon skipping ones. If you can skip the exon that has the error, then you don't have the error. But this is the concept. All right. We have, what is the evolutionary advantage of alternative splicing? Why is that beneficial to organisms?
The third one?
The third one.
Yes.
You can encode more proteins without an increase in gene number. Remember I said that a big piece of it is our ability to create proteins in response to environmental events like catching a virus. So if you have, if you can't make new proteins without gaining new genes, then you could die from an acute event. Being able to adapt to the environment is important. All right. Why might some tissues engage in more alternative splicing than others? It doesn't happen the same way in the entirety of your body.
If they need slightly different proteins product at different developmental stages?
So that is one, yes. Perhaps they need it at various stages, but they're slightly different, but not completely different. So they can just alter the proteins they already have, splice out certain exons to create the protein they want to build. What else?
If they depend on a number of related protein functions.
There you go. So again, the idea is that you can't create a completely new protein, but if you have a protein that needs this shape of key to fit in, but no, you need a slightly different shaped key over here, but it's the same class of key, that's the concept, yeah. Anything else?
It's just those two.
Okay. Yeah, I thought it was just those two, but I wanted to see if any of you guys had gotten a different answer. So I was like, I'm pretty sure it's still that two. Okay. That's good. Where are we at? Okay. So number, all right, RNA splicing. Okay, so we have more splicing activities here. Okay, this is problem, which problem is this? Oh, I forget which one it is. We're just at 1224, okay. The 1224, there was, you know, you guys spent a little bit of time on it. Obviously, there were some ones where you had to pick the correct ones, and it would give you the wrong answer if you picked mostly correct, but not all of them. So, but just walking through them and making sure you understood that. The next one you guys did pretty good on. So I guess we get into one of them that you struggled on a little bit at the end here. Okay. Characteristic of RNA splicing in eukaryotes. You can tell me which one. We can view the available hints. That's fun. Oh, hint one. Okay. Which one is this?
It's the second option.
Second option. Okay. Talked about those small, those tiny NRAs, RNAs that recognize the 5G splice junction, 3AG splice junction, and they attach on there. Those little U1 and U2, the S and RNPs, right? SNRPs. Okay. Splice junctions by the spliceosome. S in RNP is best described as
small RNAs associated with protein complexes in the nucleus.
yeah, so they're tiny RNAs and they associate with the protein complexes, allowing them to recognize the splice sites. Again, if the splice sites are broken, they aren't clearly marked, then bad alternative splicing can occur, leaving introns in. But this should be where it is. Again, sometimes other proteins will attach to do the alternative splicing. All right, and then which of the following is most likely attributed to a base substitution of the 5-splice junction. Which one is this?
I think is an amino acid substitution in the final protein product.
Okay.
I think.
So I think that's not right.
Yeah.
I was thinking it's actually one of these two.
It's
That's
a
even
longer than
usual.
usual final transcript.
Okay.
Okay.
The key here is that this amino acid substitution, generally, if it's a splice junction, it's not going to be, if it was broken, it would actually just be a longer than usual final transcript, and it would have a string of amino acids that were added instead because an intron will be left in. So this isn't quite right. It's going to be the longer t' usual. Okay. And the reason it's longer than usual, again, you could block intron removal if it didn't recognize the splice site. Even if the three prime splice site was in, it wouldn't cut out the intron properly. So you wouldn't get that loop, that cut off. All right. Viral genomes and eukaryotic genomes are similar in which ways?
They both have the ability to pack a genome into a small value.
Right.
Volume.
Viruses and bacteria and that they all have the ability to basically that complex winding, right? We talked about the negative versus positive supercoiling. All right, bacterial chromosomes. Remember, we're talking about chromosomes. We're not talking about the, we're talking about bacteria, not viruses too. This would not be the COVID.
Double-stranded circular DNA.
Yeah, they're circular DNA. That's right. Okay. And that changes the way that they divide as well. All right. Okay, polytene chromosomes. Okay, so this is, I'm gonna guess one of the ones that, oh no, you guys pretty good on this one. Never mind. It's the next one that you had trouble with. All right. So I see that most of you guys did well, but the concept here is that you have the, from numerous, what?
DNA replications.
Okay, so numerous DNA replications, pairing of homologs, and absence of, oh, sorry, this is cytoplasmic division. Absence of strand separation or cytoplasmic division. That should be the concept here, okay? Not leaving the chromosome. It's not dividing in mitosis. This is constantly doing DNA synthesis in a single chromosome and not dividing up. Each chromosome contains about, so you'd have to look this up. I forget the number I gave you. So
1,000 or 5,000.
okay, 1,000 to 5,000. There you go. This did seem a little high. All right, they appear in specific tissues. What was the tissue I told you it appeared in?
Salivary glands.
Yeah, so this is because they have to create so much incredibly dense amounts of saliva to go into their pupal stage. Okay. Dipterans such as, the only dipteran we've talked about, Drosophila. Okay. So they have these sharp, light and dark sections along their length. Such bands, known as, well, there's not a lot of abilities here, right? Obviously, it's not 5,000 to 10,000. That makes no sense. Or that doesn't make sense either. So it is going to be the chromomeres. Okay, good. Oh, that's right. All right. We had the telomeres part. You could tell me about telomeres. Ebery, you've already answered a bunch of questions. Someone hasn't answered yet. Come on, you guys remember telomeres. There's a whole little read up on telomeres activity. Which is false?
The first one.
Okay. So they are indeed related to the structural integrity of chromosomes, right? It keeps them from degrading over time. And they would stay more darkly because they are made of heterochromatin. And they replicate late because the replication has to do with this is the last part, and they don't do anything genetically. All right, so it looks like that was the one that you guys actually got wrong. And so I think the key is if you walk through the different things that they do, you can see that they are related to certain things because we know they are heterochromatin. The reason they replicate later in the S phase is that they are at the end of chromosomes. They are genetically inert. And so the stain more lightly, that's the density of them, okay? Basically, you can't see them as well because they are that inert part. All right. Okay, so this is just a coding piece. So you had to be able to use this kind of setup. This would be first base, second base, third base, sort of like a little puzzle you have to work through. Okay, I will tell you guys, you will see something like this on the test if you haven't already taken it. So just know that you may have to do, this is good practice. I just want you to be able to understand how to do that, okay? All right. And then you would drag them into various tiles, right? Oh, I forget which one, the UAA and UAG and UGA. UAG, there's no UAA, right? Oh no, wait. There's UAG, okay. Challenging myself here. Oh, there's UGA, that's also subcode. So again, these kind of pieces is something you should be able to do. And you know, this one is also an amino acid, okay? That's how you put them all together. And so you need to be able to read this. And one of the key things here as well is I want you guys to understand that when a stop says stop, you stop, okay? So you'd need to fill these out in this way, okay? You have to go back and look up. These kind of tables are sort of everywhere. So I would say that the tables like this, don't stress out too much about that because you should be able to pull one up. I think I actually have one in my lecture too. So, but that's it. You would go AUG, that's obviously methionine, GCA, AGA, AAA. Okay, all those options. Okay, I'm not gonna do this one because I would have to go up and down like a million times, but with a little bit of work, you can get through this. Okay, looks like you guys did get through that just fine. Does anyone have questions about how to read this or do it? Do you want me to go through it? Okay, then I think we're good. All right. And this was sort of a gimme, right? In paramecium, the code on UAG encodes glutamine. What does it encode in humans?
Start. Oh, stop.
Yes, stop.
Stop. Stop.
So if you just went through that, you'd remember it was a stop codon. Again, stop codons are different in different organisms. For most eukaryotes, is those, and those are the ones that we normally use. But paramecium uses a different stop codon. Okay. So for that one, that was just kind of the end of the chapter 12 and some of chapter 11 that we covered. You know, there will be a little bit on chapter 13 that I finished up on Tuesday, but don't stress out about that one too much. Again, mostly what you saw in the homework for the building the amino acids is mostly what I want you to know. How do you build amino acids, and what does it mean when it's long here, but what about stop codons and how does that affect the protein and such? Okay, so being able to interpret those pieces and read the charts, but I didn't put a whole lot about translation because the main focus of this last piece was how does our body interpret mRNA? How does our body regulate genes in terms of the DNA structure, chromatin structure, and RNA processing? And how do errors in any of those impact gene expression? All of that's connected to a lot of the pieces we've been putting together. So now we've moved away from just how do we inherit genes to now how does our body interpret genes? How do we actually get genes to express to ourselves? All right. What else? If anything you guys have questions about, I'm available right now. Your test is up, been up since yesterday. You have until Saturday to take it. Take as much time as you want. You guys can all take it at once, as far as I know. I don't think there's any limitation on that. So technically the availability date ends on Sunday, so you could take it until about 2 a.m. on Sunday, but it's going to mark it late for me. I'm not going to, as long as you were able to start it, I'm not going to mark it too late. You do have to make sure that you go and you have to make sure that you post the honor code to there. Make sure you read through the honor code. You have a choice of either writing down your name and saying I agree with the honor code, or typing your name in and submitting the honor code. Either way is fine, as long as you have a submission of some sort and it says your name on it. That's my preference. That you are certifying that you've read it and you agree to participate in this honor code. Basic concepts of the honor code. I just don't want you guys to look stuff up on the internet. But if you go to the class page and download the PowerPoint that I had on a subject and read through one of the slides, that doesn't count as accessing the internet. That's fine if you just don't store them on your computer. You're going to read them. You know, you have them on Google Docs or something. You have to access Google Docs. The key here is I just don't want you going into any external sites to access outside information, to access type in this information and you get the gene link. That's not what I want from you guys. Try to be honest about how you do it. Also, if you're able to, in some parts, I give you a segment to explain what you did, and this will hopefully help you to get partial credit. So if you can't explain what you did because you typed it into a calculator, you may actually get the wrong answer. So, all right.
Professor? I have two questions. One would be, well, I know the test is timed like an hour and a half, but what exactly was the final formatting like?
Okay, so it's going to auto submit. So an hour and a half, you'll have to keep track of your own time, because I could not figure out a way to make it fair to you guys. One of the things I'm trying to avoid here is either you forget to submit it and then it marks it late or extended time, or you you take like a day or two to complete it and submit it, even though technically you were only supposed to use an hour and a half. So it is going to submit it. It submits it. It gives you a 5-minute grace period. Just I suggest you do the test as if it's going to give you 90 minutes. So you have a little bit of buffer so it doesn't submit it right when you're typing the last problem, okay? But it gives you a 5-minute buffer past that. You have 95 total minutes, but treat it as an hour and a half, and then that's the concept. The format overall, it's a mixture. I have calculation, fill in the blank questions, similar to ones that you've done on through the the homework and such. Type in the length of this, type in the length of that, whatever. Based on information that I give you, and I may give you the length, and then you have to figure something else out. I may give you just the results. So, and I do have them cycling through. So for each calculation question, there's at least 3 or 4 others that will cycle, and they're the same concepts. You just have to answer a different question about them. But it took me so long to build your test. But it's good. There's a few extra credit questions in there as well. I basically built in about 6 extra points. So I think the extra credit questions, if they're marked, they may be random questions. They weren't true extra credit questions. I just put extra questions in there. So if you answer all of them correctly, then you have more points.
Okay, thank you.
Anyone else have any questions? All right, so I will remind you that in 2 weeks when class ends, that's also when you're the end of the last week of classes before finals week is going to be the day you have to turn in your research paper. I will not be able to grade your research paper beforehand. Remember that after you take the test, you will have you'll have to change the grade tool a little bit if you want to work the grade tool after that. Basically, just put in your total value after the test is graded, and then take that segment out that says that's left over. You would just delete the exam 2 out of your options. Anything that you've already been graded on, you delete off of the grade tool. So that's why I marked it as being for April 6th in particular, because as I grade things, the number of available points goes down, and hopefully your actual score is going up. So again, it's going to be about 200 points you don't have all the information on, because it's going to be the final, which is a bit bigger, and the paper. So you don't have all the information, but you should have an idea of where you're going based on previous tests, because the final is comprehensive, and there's only a couple more chapters that we're covering outside of other exams that tested you.
Oh, I forgot one more question. For the extra credit journals that are due tomorrow, can we email you the link of like the article?
I want everyone to know that I was supposed to switch that when we first restarted. They're due next week. You're fine.
Oh, okay. Thank you.
I apologize that I didn't put an announcement up. I realized I hadn't changed them, and I changed everybody's because I have like 3 classes that have that. So I was like, okay, everyone's are changed, but I forgot to tell everyone. So if you turn it in tomorrow, that's fine. If you're ready to turn it in, good for you. But you have another week. So if your score on the exam spurs you to do it instead of you were lackadaisical about it before, then that's fine. But yeah, so it's not due tomorrow. But you had a question about it in general?
Yeah, when we find an article that we want to write it, can we email it to you to see if you approve it? Okay.
Absolutely. I'm happy to tell you, yes, good article. That's basically that would be my response. It was a good one. Or if I think that it needs to be improved, like I'm like, oh, this is actually a blog. Oh, this is from a science article accumulator. Try to use the original source. That's probably my comments on it. All right.
Professor, can you repeat what you said earlier about the research paper?
Research paper is due on May 1st. Basic idea. Okay, so that's the last day of classes. Also the day that you, the last day for anyone to drop from the class and get a W. Okay, so as I explained on Tuesday, this is a stressful time for everybody. So if there is something that is preventing you in some way from completing the class, if you have more than 70% in the class at the point of the drop date, even if you haven't finished the last two assignments, which would be the research paper and the final, then if there is something that's preventing you from finishing the semester, incompletes are available if it is something unavoidable or something that's related to an issue you're having. We're going to be liberal with the incompletes. Do try to complete it because incompletes will delay your credits, but please don't hesitate to talk to me if you are actually struggling and are unable to complete any of the projects. And if that's true, I cannot extend any time to you because there's requirements about when I'm allowed to put my grade out. But what I can do is give you a incomplete, and you can finish that by the fall semester. In particular, since the final is going to be online and the research paper is just emailed, those last two are flexible, but only if you take it incomplete. So that's the information there. And it would require you to have a passing grade already, so you can't just use it as a delaying tactic. And again, if you access the exam, this has happened to a couple of my students. If you access the exam and you have your computer shuts down, someone took a picture of their computer and sent it to me. They were like, it's got a blue screen. So if your computer shuts down, if your internet cuts out and it says that you had your last activity, just send me an email right away and I'll check. I'll just check to see that, oh, you did only finish half and then suddenly stopped, and I can see how much time you spent in it. So if you obviously stopped before it cut you off and you didn't finish it, then I can see that was unintentional. I can get you back in. Okay, so just let me know if that happens, because I do only have one chance for everybody. I'm assuming at this point that you have reliable enough internet to sit down and take the test in one go. But if something happens, then just let me know, because my internet was going in and out earlier today. So it will not penalize you for, the only thing I won't allow is for you to retake any part of the test. So someone had emailed me about a different test in a different class and said, can I have just 10 more minutes? I'm not going to give you 10 more minutes. Okay, it already has a little bit of a buffer worked into there. You don't get 10 more minutes, but you are able to tell me if it cuts you off prematurely, and I'll be able to see that. Okay. And I'm posting the case study 10 right after this. So I'm going to work on all the postings for this class. I apologize for not getting back to any of your previous class activities. I've been very swamped with test creation. So tomorrow I'm going to work through all of the piles of ungraded things that have been graded in the past week. So you'll see that pop up hopefully before the test is finished and graded. Good? Thumbs up? Stop my share for now. I need to stare at that screen. Okay, I see thumbs up popping across the screen. You can go down to reactions and give me a thumbs up. Excellent, okay. All right. Well, I'm gonna post those things and then I'm gonna go have a not working night and I'll do my grading tomorrow, okay? Everyone have a good night. Yeah, I'll talk to you next week. Please let me know if you have any issues with the exam. We'll fix that.
Bye.
Bye. See you later.
Thank you, Professor.
You're welcome.
Bye, Professor.
Bye.
Bye, Professor.
Bye.
That's